All right, ladies and gents, so welcome back. We're going to continue our discussion of non-Mendelian inheritance traits by talking about sex-linked traits today. So first and foremost, take a step back and remember your chromosomes, right? So when we look at a karyotype in a human, we're talking about 1 through 22 that are your autosomes. So that's where the vast majority of your genetic information is stored. But your 23rd pair is what ultimately determines whether you will be male or female. And not only does your X and Y chromosome contain the information to determine gender, but particularly your X chromosome also contains additional information. So when we look at going through meiosis, splitting up those chromosomes, ultimately, who decides whether or not that baby will be a boy or a girl? Well, remember, it's ultimately dad's information that determines that. So mom is XX, so when her cells split out, all she ends up with are X's. Dad's, on the other hand, is X and Y, so dad could potentially pass on an X, match that with mom's X, and you get a daughter. But if dad passes on a Y, along with mom's X, you end up with a boy. So when we look at sex-linked traits, these are traits that could potentially be connected to gender determination, but more often than not, when we're talking about sex-linked characteristics in genetics, we're talking about additional characteristics that are also carried on these sex chromosomes. So who do we have to thank for understanding that this even exists? We'll go back to that very first slide, and I mentioned a gentleman named Thomas Hunt Morgan, and he is the one who is given credit for discovering the fact that traits exist on the sex chromosomes that go above and beyond just gender determination, and he figured this out in fruit flies. What we do understand now is that those genes that are attached to your X and Y chromosome, many of those on the Y chromosome do really connect to male-specific traits. But if I look at the X chromosome, there's about 1,100 known genes, including a lot of genes that we know have genetic disorders associated with, particularly oftentimes their recessive counterparts. Your Y chromosome, on the other hand, considerably smaller, about a third of the size, and only has about 250 known genes, most of which are male-specific traits. So why do we care? Well, when we look at sex-linked traits, first and foremost, most of the harmful alleles that we see, when I go back here and take a look at things like the muscular dystrophy, the X-linked deafness, the hemophilia, for example, most of those are recessive, meaning that you would need to inherit two recessive characteristics or two recessive alleles in those traits if we were typically talking about traditional Mendelian genetics. But here's the thing. In females, they're the ones who get those two X's. So they are actually capable of being that traditional heterozygote individual where they have an X that is dominant, an X that is recessive, and as a result, that dominant X can mask the recessive one. So they can be those traditional carriers that we have mentioned before. But here's where it gets interesting, and this is what Thomas Hunt Morgan was given the Nobel Prize in Medicine for. And that is because most sex-linked traits seem to disproportionately affect males above females. So far more males get affected by sex-linked characteristics than females do. And there's a really good reason for it. So while females carry two X's and have the capacity to be a heterozygote or be a carrier for these recessive traits, males don't. So males will express the trait anytime that they get an X that is carrying a particular recessive trait. Because that Y chromosome is tiny, contains very few genes, and doesn't have the capacity to mask what the X is carrying. They're not a one-to-one match like every one of your autosomes is. So males don't have that second X copy to mask a recessive characteristic. As a result, if they inherit an X with that recessive trait, they are going to show it. So here's the example in fruit flies. This is what Morgan figured out. What he was dealing with wasn't necessarily recessive disorders, but just recessive traits in general. And so when you look at eye color, for example, in fruit flies, typically the sort of natural, what they would call wild type or dominant type, is red. And so he was crossing fruit flies, purebred reds in his females, purebred whites in his males, mated them together, got that F1 generation. Nothing looked unusual. They were all red as he would have expected. What became interesting, though, was when he took those heterozygotes or those F1 individuals and mated them together, he saw something interesting in his F2. He got a 3-to-1 ratio, which that in and of itself is not necessarily the interesting part, but what he noticed was that almost all of the white-eyed individuals were males. So overwhelmingly, his white-eyed individuals were males, not females. And that the red eyes were the female instead. And so that's what started him questioning where was this red eye trait located. And what he eventually discovered was that it is in fact on the X chromosome. So by mating these individuals together, that male doesn't have that second X to cover up a recessive trait. And so overwhelmingly, the white-eyed individuals happen to be male. So the disorders that we see associated with sex-linked traits are considerably more common in men who lack that second X to cover it. So we look at things like red-green color blindness or hemophilia or male pattern baldness. These are all examples of sex-linked disorders that disproportionately affect males above females. It's not that females cannot have this disorder, it's just that she would then need to inherit both a recessive trait from her father as well as a recessive trait from her mother, where males only need to inherit that recessive trait from their mother, and that's it. So the likelihood of a male expressing this trait is much higher than it would be in a female. So just a quick colorblind test. If you can see all of these different letters and numbers, or at least the vast majority of them, then good news, you don't have red-green color blindness. But if you can't pick out the numbers in any of these, then perhaps that is something that you probably at this point in your life already knew, but it's just kind of an interesting quick test. So let's do a couple of examples here. So if I look at some examples with baldness, all right, mom is going to be a carrier, dad is going to not be bald, so he is not going to have this trait. So when we write out the genotypes and do the Punnett squares for sex-linked traits, there's a couple of new conventions here. First and foremost, I have to recognize and utilize the X and the Y chromosomes, and then I will use that more traditional capital versus lowercase dominant recessive traits that are attached to those things. The other piece to recognize here is that there is nothing attached to the Y chromosome. This is only X. So if I take a look, mom's a carrier. So here are her two X's to designate that she's female, and she is a carrier for it. So she's got a capital and a lowercase. Dad is not bald, so he has an X and a Y to distinguish him as male. And then he is not bald, so he has the dominant trait on that X. And then you do your Punnett square as normal. So when I go and look at the phenotypic ratios, I can write these out in a handful of ways. I'm going to be on the order of overly specific. Depending on the way questions ask you, you may or may not have to do this, so just read the questions carefully. But what I end up with here in a situation where I do have a 3-to-1 ratio, 3 who are not bald to 1 who is recessive and is. But if I start splitting out what I look at between males versus females, all right, I have 100% female who are not bald, and then I have a 50-50 split between my males who are bald and who are not. I can also indicate the fact that 25% of my females are also carriers versus 25% of my females who are not carriers. So these ladies will never pass that bald trait on to their children, but these ladies do have a potential to pass it on given the fact that they are then carriers for it. Another example, So here now mom is not a carrier, so she's got two capital B's, but dad is bald, so dad's got that lowercase b on his X. Interesting thing here, all right, all they produce are carrier females, but none of their men are bald, all right? So think about how that's going to have to work. In order for those boys to show any sort of bald trait, it always ends up coming from their mother, whether she is homozygous or heterozygous for it. If she doesn't carry the trait, then there's no way for her sons to end up with that X-linked characteristic. Dad, on the other hand, can determine whether or not his daughters have it or whether or not his daughters are carriers for it, but that is all he is influencing in terms of the trait itself. What he passes on is strictly the Ys to determine the males, not whether or not they carry a sex-linked characteristic. Something else that's interesting while we're on the topic of sex chromosomes that occurs in females is this idea of X chromosome inactivation. Now, this only occurs in females because they have a second X. In order for an organism to survive, they only require a single X. So, as a female having two X's, one of her X chromosomes gets randomly deactivated or shut down or condensed during embryonic development, and it's random from one cell to the next. So, if I were to go up to a female and take out a cell from her calf, all right, and look at which X chromosome was shut down, it may look very different from the X that I pull out of her fingers or out of her skull. So all of those X chromosomes that do get deactivated, they get condensed. We call them bar bodies. The one that is uncondensed is the one that's being expressed. And again, not every X gets deactivated in the same cell everywhere. So if I look at individual cells, I can talk about which X gets shut down. But if I look at her as an organism as a whole, she still expresses both X chromosomes. So why is this kind of an interesting scenario? Well, for most female human traits, it's not something we can physically look at you and know. But I look at cocalico cats, on the other hand, and I can. So when we look at cat color in terms of this orange versus black, it's actually carried on their X chromosome. So if I look at a cocalico cat, in our case of a cute little orange male here, he happened to inherit the orange X and the Y, which made him male. But for all the females, they've got a black, or an orange and a black. So depending on the patches of fur that actually shut down the different chromosomes is where you get the pattern from. So if I were to look here, all right, here all of her cells in this patch here allowed the X that was black to be expressed and shut down the orange. I come down here and the opposite's true. The orange is now expressed and the black was the one that was deactivated. So because males can only have one X chromosome, I can't shut down any of their X's. I can't do this X chromosome inactivation, and so you'll never have that cocalico coloring in a male. It's female only. All right, if I were to try to shut down that active X in a male, it would be lethal to that cell, and if I tried to do it to the organism as a whole, all right, it would be lethal to the organism overall. So males cannot have cocalico coloring because they can't do this inactivation of their second X chromosome because they don't have a second X chromosome. So just kind of an interesting thing. So anytime you see a cocalico cat, you automatically know that it's female. One last thing that I wanted to point out before we move on from these exceptions to Mendel's inheritance is just what makes the organism what it is. And there's been this sort of existential debate for a long time about how much of your genes are determined by nature versus nurture, how much is coded for in your genes versus how much is influenced by your environment. And truly, all inherited information is overwhelmingly coded for in your A's, T's, C's, and G's. It's coded for in your genetics. All right. So any characteristics that are acquired through environmental influences, more often than not, don't influence your genetics as a whole. So it's generally not something you're going to pass on to your kids. That being said, remember when we talked about your epigenome, right? So your epigenetics, these tags and these condensing and relaxing of certain parts of your genome. So there is a lot more to it when we start digging into that level of your genes. So to truly say we talk about nature versus nurture and it's either this or that, the reality is that it's a lot of both. There's definitely your genes and that DNA sequence that you get inherited from your parents that influences your phenotype and the way that you look. That being said, we also are starting to understand more and more about how nature ends up influencing how that gene gets expressed, when it gets expressed, to what degree it gets expressed. So in reality, it's probably a little of both. It's the question of then what trait specifically are you talking about, and how strongly is that particular characteristic influenced by one or the other.